afternoon, everyone. I want to start 1 minute early because I want to make sure the sound is correct. Can people on Zoom hear? You can, maybe one person could unmute and say yes, and we'll test the sound check back.
Yes.
Yes. Yay, thank you. Okay, and we can hear you. So make sure you're on mute because the whole room can hear you. All right. Welcome to the Fall 2021 Hybrid EEB Seminar Series. I first of all want to thank Bea Bazelga for leading the way in organizing the seminar series this semester, in addition to Ruth Valesky, Christina Kramer, and Neil Jaron for logistical, technical, and physiological support in providing refreshments afterwards. And I want to thank everybody for coming in person who can make it in person, and everybody who's on Zoom for coming via Zoom. So for this semester, we have prioritized local speakers for the first half of the semester, so we can have hybrid seminars, making a Zoom link available for those off campus, but also encouraging people to reconnect in person when possible. We will have some to-go outdoor refreshments afterwards. And today we're going to kick off the seminar series with our very own Ford Dennison. Ford did his graduate and postgraduate training at Cornell, UC Davis, and UCLA. He was a professor at UC Davis and director of their long-term agricultural research for a little over a decade before starting as an adjunct professor at EEB in 2005, and shortly thereafter CFAN's fellow. He's continued in his adjunct role since 2005. Ford is well known for his research on the evolution of mutualisms and what this means for applications in agriculture, especially around nitrogen inputs. He also co-invented devices to assess root nodule function and measure sunlight capture by field crops. Ford has written a highly regarded book, Darwinian Agriculture, How Understanding Evolution Can Improve Agriculture, which, as stated by one reviewer, is a brilliant book by a terrific scientist. So let's welcome Ford here to start off the return of our seminars to campus. Thank you, Ford.
Well, I'm having my doubts about heterosis, otherwise known as hybrid vigor, but there are advantages to the hybrid format, so that's what we're going to do. So I've been studying nitrogen fixation off and on for 45 years. I will talk mostly about the last 20. And the long-term goal of this research has been to find ways to reduce the need for nitrogen fertilizer without reducing crop yields in a way that would promote, say, like if we could just ban fertilizer, right? And Brazil would be happy to grow the corn that we would not be growing by clearing the Amazon. That's not the approach that I advocate. Let's see, how do I get my next slide? Just a minute. I'm going to stop sharing momentarily and fix this. At least I hope I am. Yes. All right. Is that working in Zoom land as well?
Yes, it is.
Okay. So I'm going to argue that we could cut fertilizer needs in half by breeding for lower protein cereal grains. We would then need to replace the missing protein, which I'm arguing we could do with nitrogen fixing grain legumes, but only if we can increase the amount of nitrogen that they get from symbiosis without increasing the cost to the plant. I'm going to suggest that we could do that by taking the existing host-imposed sanctions against rhizobia that fail to fix nitrogen and breeding for stricter versions of those sanctions. And then at the end of the talk, I'm going to talk about mutual manipulation of host and symbiont in this symbiosis. Suggest that it's generally harmful to our interests at least, but that there may be cases where it could actually be beneficial. So a lot of us work on natural ecosystems where nutrient cycling within a fairly closed system is typical. And so you need to expand your worldview and think about agricultural systems where we insist on large amounts, we need large amounts of nutrients to be exported to cities, like Twin Cities. And so somehow those nutrients need to be replaced to maintain any kind of a balance. This is typically done for corn with fertilizer, but I'm going to argue that we can rely instead on nitrogen fixation. Here's the reaction: nitrogen gas, energy used by the bacteria using resources provided by the plant. Ammonia is produced, which is then taken up by the plant and used to make protein. And there's hydrogen gas as a byproduct, which will turn out to be important to my analysis. Now, usually when people think about relying more on nitrogen fixation, they think about the crops in rotation with maize, for example. Soybean can get most of its nitrogen from symbiosis. But if you actually do a nitrogen budget for these systems, it often appears that there's actually, well, basically the nitrogen that's taken up by fixation is somewhat less than is actually exported in the grain. So this doesn't have a surplus to contribute to the maize. Instead, often in modern agriculture, there's a bit of fertilizer carryover that's subsidizing the soybean. So that's one problem. No surplus to transfer to the crops that can't fix their own nitrogen. But the other problem is that during the months between when the soybean is harvested and the maize is grown, that nitrogen in the soil can, even though it originally came from biological fixation, there's nothing that keeps it from leaching and polluting the groundwater and destroying contributing to the dead zone and so on. And an example of that from work that we did back at Davis in Davis, California, you can grow a cover crop during the winter when the crop wouldn't necessarily be grown normally. This is an early stage in the growth. By the time it's ready to plow down, it's actually produced enough nitrogen to be a significant contributor to the following crops. But the release of nitrogen from this decaying material is not automatically synchronized with the uptake by the crop. And so Hiroshi Hasegawa, who was younger when this work was done, ran a well-regarded model. It doesn't mean the model is necessarily right, but compared the amount of leaching of nitrogen from this cover crop before it could be taken up by the wheat crop relative to how much leaching you would get from well-managed nitrogen fertilizer, and concluded that you could actually get more nitrogen leaching from the cover crop. Now that's dependent on the fertilizer being well managed, which of course is not always the case. But nonetheless, this reliance on another legume in a rotation to feed crops that aren't able to fix their own nitrogen is problematic. And so I'm going to suggest another approach. Aren't I? I think I am. Yes. Which is certainly speculative, but this is an idea I stole from Adam Smith, whose picture is hidden up here behind mine, Wealth of Nations, where he argued that much of our progress over the course of civilization has come from increased specialization. If you specialize on something, you can get really good at it. And he was talking about humans, but it occurred to me that that could perhaps also be applied to plants. And so maize is an example of a plant with the C4 photosynthetic pathway. They have these little compartments where they concentrate CO2 that has two benefits. First of all, it gives them a very high photosynthesis rate. And second, because the photosynthetic enzyme is the main nitrogen component of the leaf, they can actually have high photosynthesis rates without very much nitrogen in their leaves. But we know that corn needs a lot of nitrogen, but that's because of all the nitrogen that's being exported in the corn grain. It's not just for photosynthesis and vegetative growth. Now let me explain this graph here, which will illustrate the changes that I'm proposing. This horizontal axis is the metabolic cost of making seed for each of these species. And the metabolic cost of making protein and oil is about the same. Starch is cheaper, but protein and oil are about the same. So we can actually move up or down in this graph, changing seed composition without expecting, at least in theory, without a change in the total amount of crop that's produced. Okay. And so what I'm suggesting, since the reason we have to fertilize maize is to replace all the nitrogen that's being exported in the grain, cut the nitrogen, that's this axis here, the protein content, cut the protein content of the grain in half, make the same amount of corn, but mostly more oil and less protein. Oil doesn't have any nitrogen, it doesn't need any fertilizer to replace it when it's harvested. And because maize is so good at photosynthesis, and that's what oil needs, basically, is carbon, we could have the same yield, but half the fertilizer. So that's basically what I'm proposing here, cutting fertilizer in half because we're only exporting half as much nitrogen in the grain. But that leaves us with less protein. So the other piece of this division of labor is that legumes like soybean can get all their nitrogen from the air, no nitrogen fertilizer required, because of their symbiosis with root nodule bacteria. And so we go up here to the soybean, we can do the same thing. Moving in the vertical direction doesn't change the yield because the glucose cost is the same for oil and protein. We get more protein from the soybean to replace what we're not getting from the corn. And this is what it looks like. The same total protein and oil yield, but half the fertilizer. This, of course, is in addition to all the other things that we're doing in terms of, putting fertilizer on at the right time and not putting on more than is needed and so on. It's on top of that, we can cut it in half, I think. But that assumes that we can get all that, we can get that increase in nitrogen fixation without having to divert a lot of metabolic energy more than we're spending currently. Because if we did that, we wouldn't get what we're after. Now we get to the evolution. So my argument is that in order to get more nitrogen fixation without having an increased cost to the plant, we're going to need better cooperation between the two partners in this symbiosis. And so that seems to be in conflict with the other well-known idea from the Wealth of Nations, namely that there's this magic invisible hand where individuals pursuing their own interest benefit society as a whole. Again, he was talking about humans, but that's actually the way a lot of people have thought about symbiosis, prior to about 2000. That either that symbionts were all altruists, or that somehow just doing their thing would be best for everybody. And that's not what my research suggests. And in fact, if you read The Wealth of Nations in any detail, he gives lots of exceptions to this general principle, exceptions which are not mentioned by people who think we should do away with taxes and regulation and so on. So this idea that, yes, sometimes individual selfishness, whether it's a human or a plant, may benefit the whole community, but not always. The theme of the tragedy of the commons, explored in much more detail by Eleanor Ostrom, who won a Nobel Prize for her work on how people manage those kinds of challenges. And Robert Frank has written a number of interesting books and op-eds in the New York Times on this topic, which I recommend. But that's all humans, and the rest of the talk will be about bacteria and plants. So is there a conflict of interest between the legume plants and the bacteria in their root nodules? Well, there is, and here's one example. So this is kind of a cartoon of what's going on. The plant is supplying carbon. Ideally, from the viewpoint of the plant, the bacteria should respire all that carbon to CO2, use the ATP from that respiration to reduce nitrogen gas to ammonia gas, give almost all of that to the plant to make proteins. But most bacteria, most nitrogen-fixing bacteria in this symbiosis, divert a significant amount of carbon to this lipid called PHB, or polyhydroxybutyrate. Now, as you would expect, diverting energy from nitrogen fixation means less nitrogen fixation. And indeed, Symbiosis et al. Compared, this is nitrogen accumulated per plant over the growth of the plants. And so he compared the wild type bacteria that do divert resources to PHB with a knockout that he made. And sure enough, if you get rid of that diversion, you get more nitrogen fixation. So from the plant's point of view, this is bad. On the other hand, the bacteria are doing it for their own reasons. And Catherine Mueller, now working for USDA, but it was a recent PhD student with me, did some modeling of the benefits to rhizobia of accumulating this PHB. That work is published, and what she predicted from the model is that with a plausible level of dormancy on the part of the bacteria, if they're not really actively running around, plausible level of dormancy, and the levels of PHB per cell that she measured in rhizobia from root nodules, that PHB could last for years, keeping those bacteria alive in the soil until they can find another host. Well, in this unpublished data, which we submitted recently, she actually tested that hypothesis by going down to our field station in Waseca, where there are plots where soybean has been grown every year for the last 30 years, and plots where soybean was grown 30 years ago, and then not again since then. It's been, I think, continuous corn for the last 30 years. And so what she did was to take rhizobia from these different soils and did a common garden experiment where she measured how much PHB each of these populations accumulated. And so what she found is that the strains, it's hiding behind here, the strains that survive for 30 years without a host actually, in a common garden experiment, accumulated more than twice as much PHB in symbiosis relative to the initial population, which is what we're calling this, the ones that just came out of the soybean crop last year. So if you combine these two things, diversion of PHB is costly to the plant, but beneficial to the rhizobia. But wait a minute. I mean, the rhizobia are dependent on the plant. Making the plant grow less is not good for the rhizobia. So the rhizobia are kind of facing a trade-off potentially here. And the question is, how is that trade-off resolved? Well, quite a few years ago now, Stuart West, who's now a professor at Oxford, spent a few months in my lab and modeled this question. And so what he modeled was how much allocation of resources to nitrogen fixation is optimal in terms of rhizobial fitness. You know, what's the evolutionarily stable strategy of allocating resources to nitrogen fixation versus putting it at PHB or otherwise using it for the rhizobia's own immediate reproduction. And he modeled it first with a single genotype of rhizobia on each plant. And under those conditions, if there's not much nitrogen in the soil, those rhizobia have better fixed nitrogen, or their plant's not going to grow as much. And rhizobia on a neighboring plant that are keeping their plant growing, they're going to reproduce more. They're going to go out in the soil. They're going to be dominant in the next generation. Now, if there's more nitrogen in the soil, things change. They can get away with fixing less nitrogen, something like that. But at least with one strain per plant, a biological version of Adam Smith's invisible hand does seem to work. You know, just pursuing their self-interest is going to keep the plant alive. But as soon as we go to a realistic number of strains, so this axis is what he calls relatedness. So relatedness 1 if there's one strain per plant. And as soon as we go to a realistic number of rhizobia strains per plant, the strains that fix no nitrogen, to put everything into their own reproduction, they're predicted to win in competition against the foolish altruists that try to keep their plant alive unsuccessfully while cheaters on the same plant are putting everything into their own reproduction. Well, this is obviously wrong, because if this were true, this symbiosis would have died out 60 million minus 3 or 4 years ago, and it hasn't. So we took a look at the assumptions. We took a look at the assumptions behind the initial model and considered an alternative assumption, which was that in addition to the collective benefits shared by all the rhizobia, you know, having a healthy plant, that there would be some individual benefit to fixing nitrogen, which could come from the plant monitoring root nodules and shutting off resources or otherwise punishing root nodules that were not contributing enough nitrogen. And the plants would do this because it would save the individual plant resources. They're not doing it because of its evolutionary effect on the rhizobia, but there would be an evolutionary effect that only the more beneficial rhizobia would be reproducing in nodules and released into the soil. And so this would be analogous to, if humans do this, we call it domestication. You know, the wolf pups that bite your kid don't get to reproduce, or the you know, kale to broccoli or whatever. Genetic change in the rhizobia population imposed by their partner. Now, another thing that they might do, perhaps, is rather than, or in addition to, whatever the genotype of the rhizobia, maybe they could be manipulated by the plant in a way that would make them more beneficial. And this would be like, you know, you can maybe tame a mountain lion, perhaps, but they haven't been domesticated. Darwin wrote about domesticated rabbits. You know, the baby rabbits are friendly to people, whereas wild rabbits, they're just, you know, very afraid of people. So when he modeled that domestication hypothesis that the plants impose sanctions on non-fixing or less beneficial nodules, then over a whole range of within plant diversity, under a couple different versions of that alternate assumption, now strains that fix nitrogen have higher fitness than those that don't, because they're going to get shut down by the plant if they don't. Well, but is this true? There was some evidence in the agronomic literature that might actually be something that plants could do, but we did experiments to ask this question. And in all of these species, what we did was to take a good nitrogen fixing strain of rhizobia and in some of the nodules, expose those nodules to an atmosphere that had very little nitrogen gas, so stopping them from fixing nitrogen. Now, in theory, they're not fixing nitrogen, they've got extra resources, they ought to be able to reproduce more, but if you look at the actual number of rhizobia per nodule, we see exactly the opposite result, that the non-fixing strains reproduce less because of the plant response to their failure to fix nitrogen. And so that's consistent with the last two models. And this often showed up as an effect on nodule growth. Here you can see nodules that are fixing nitrogen. They've gotten bigger here. They have more rhizobia in them. Here, we expose these nodules to a nitrogen-free atmosphere. They stopped growing, more or less. And there were fewer rhizobia in those nodules. Well, all of the data in this graph come from cases where there was very little nitrogen provided by the rhizobia to their host plants. And I think these results, these fitness cost of not fixing nitrogen when you get punished by the plant, probably explains why it's not very common out in the field to find rhizobia that go into roots and make nodules and then don't fix nitrogen. But what is common is mediocre rhizobia that fix something on the order of half as much nitrogen as the best inoculum strains that farmers use. And so Toby Keers, who did the initial soybean work, she's now a professor at the Free University of Amsterdam, tried a range of nitrogen gas concentrations to give a range of nitrogen fixation rates. Again, this is rhizobia per nodule, and consistent with her earlier work, strains that fix no nitrogen, or these are all the same genotype, but rhizobia that fix no nitrogen because of the gas treatment had, a half or a third the reproduction of those that fixed nitrogen at high rates. But this mediocre treatment, about 4% nitrogen gas gives you about 50% nitrogen fixation. Fixing at half the rate, the plant didn't notice, didn't shut them down. They had high fitness. And we speculate that that's why these mediocre rhizobia are so abundant in the field. So going back to the applied goal of this work, we want to increase nitrogen fixation. But if you you can double nitrogen fixation by breeding for twice as many nodules. But yield goes down because those nodules have a cost. So what we're trying to do is increase nitrogen fixation without increasing the cost. And that would be a doubling in having the nodules occupied preferentially by rhizobia that are doing this rather than by rhizobia that are doing this. And just so you know, inoculation generally doesn't work. So what we need is some way that we can actually impose selection. We can, extend this domestication by the plant that the plants are already doing, but make it more extreme. And I'm suggesting this is something that plant breeders could do. So the idea here would be to start out with a genetically diverse population of whatever the legume is, measure the yield of these, but don't pick which genotypes go into the next phase of the plant breeding program just based on this yield. Because we're also looking for legacy effects that would result if, well, here's the effect. The idea, 2 years later, these are all a single test genotype of the legume. Those differences in growth are not genetic differences among these, they're legacy effects because this genotype here allowed only the very best local rhizobia to reproduce in its nodules and release those into the soil. And 2 years later, this plant is benefiting from that legacy. Now, typically there's a rotation crop, at least one. I think 2-year rotations are kind of short actually, but better than nothing. So the idea would be that this would also be a single genotype, in this case of maize. And here we can see superior growth, at least relative to these plants, perhaps because of the legacy effect of the first legumes on mycorrhizal fungi that are beneficial to maize. And this would be consistent with work that Nancy Collins Johnson did here at the U quite a few years ago. Well, let's move on. I said I was going to talk also about manipulation, perhaps analogous to taming. And I'll start first with legumes manipulating rhizobia. Who did a PhD with me here. She recently earned tenure at UC Santa Barbara. And she looked at legume species that cause rhizobia to swell up inside their root nodules to look something like that. So in the soybean nodules, the rhizobia just look like, or bean nodules, they just look like bacteria. But in a clover nodule or an alfalfa nodule or a pea nodule, there's still a population of normal looking bacteria, but the cells that are actually fixing nitrogen have these weird swollen shapes. And there are various lines of evidence that these swollen bacteria have lost the ability to reproduce. You can see that here the total number of cells and the number that can form colonies is the same number. Here, only a third, this third, is able to reproduce. But these are the nitrogen fixers. So this should remind you of bees and ants. You know, I actually I got to do an invited commentary and snuck in the word eusocial rhizobia without anybody noticing. Right. So, but one of the papers that I'm not citing today, she showed that this plant trait of causing this swelling has evolved at least 5 times. That suggested to us there's probably some benefit to the plant. And the hypothesis was, well, maybe this increases how much nitrogen the rhizobia give the plant relative to their carbon cost to the plant. And you can measure that in 2 different ways. The first one is here. This is plant growth relative to nodule weight. So making more nodules has a cost to the plant. And the cool thing that she was able to do was to find a couple different examples where one strain of rhizobia goes into both kinds of hosts. It's swollen in one host and non-swollen in another. And in both of those cases, she found that the swollen bacteria gave more growth relative to nodule weight relative to the same genotype of rhizobia when they were not swollen. So there's one benefit to swelling. Now, if anybody's done this, picking all the nodules off of a plant, separating it from soil and all that, and weighing it is a lot of work. And so people don't really like to do that 3 times a week to, follow nodule growth over weeks. But that's what I'm doing, because I've programmed this little Raspberry Pi here to take pictures of a backlit root system that looks like this. And then we feed that to an artificial intelligence program, which identifies all the nodules and measures their sizes. And we can add that all up and get nodule mass per plant 3 times a week without much work. But going back to Ryoko Ono's work, the other cost is a cost of making nodules, but there's also a cost of operating nodules. And that cost of operating nodules is reflected in respiration of the nodules, CO2 coming off the nodules. And so Jeff Harms, who has a shop down in the basement of BioSci, came up with this really clever system where we have this little gasket here that turns one of these growth pouches into a cuvette where I can do gas exchange and measure respiration as CO2, but also hydrogen, which I mentioned earlier is a byproduct of nitrogen fixation. So from those 2 things, we get the nitrogen fixation rate and the respiration rate. But the respiration is the whole root. It's not just the nodules. We're really interested in, the cost of fixation, the respiration cost of fixation. So to do that, I'm building on this insight of the late John Whitty, who I got to spend a month with in Wales quite a few years ago now. And what he realized is that the nodule interior, where nitrogen fixation is taking place, is actually oxygen limited, in the sense that if you raise the oxygen level a little bit, if you raise it a lot, you fry the enzyme and lose everything. But if you raise it a little bit, you get more respiration, more ATP, and more nitrogen fixation. Whereas the root is oxygen saturated. Changing the oxygen a little bit doesn't change the root respiration. So, I don't know, did Adam Smith invent marginal gain as well? The marginal increase in nitrogen fixation with an increase in respiration, the slope of this line is the efficiency of operating the root nodules. And there too, the same strain of Rhizobia was more efficient, producing more nitrogen relative to its carbon cost when it was swollen relative to non-swollen. These two examples that Ryoko was able to study. Well, that's manipulation of Rhizobia by plants. I'll briefly talk about the flip side of this, which is manipulation of plants by Rhizobia. Now, it's been known for a long time that many Rhizobia produce plant hormones, or things that are so similar to plant hormones, you might as well call them plant hormones. And this hasn't been studied very much, but there are a few cases, and this is one of them. Will Ratcliffe, who's now a professor at Georgia Tech, did a PhD with me about the same time as Ryoko. This is a case where, rather than making a plant hormone, the Rhizobia actually make a toxin that blocks the operation of a plant hormone. It interferes with the plant hormone ethylene. And as you would, I hope, expect, this manipulation of the plant by the Rhizobia for the benefit of the Rhizobia has a cost to the plant. Here's the wild type manipulating strain, plant growth, and here's growth of the plant when using a knockout that doesn't interfere with ethylene signaling. Well, okay, but presumably the Rhizobia do this because it's benefiting Rhizobia fitness. And so Will took a look at that and first did an obvious measurement of Rhizobia per nodule and really didn't see a significant benefit to the manipulating Rhizobia in terms of Rhizobia per nodule. But he was also able using the flow cytometer in Tony Dean's lab, to measure PHB per cell. And there's a big difference that these Rhizobia that were manipulating the plant, those Rhizobia were accumulating a lot of this PHB, which Catherine Mueller showed could really enhance their survival in the soil. Will actually did some survival work as well that was consistent with that. Here's a more recent example of rhizobia manipulating plants. In this case, they are making a plant hormone, the hormone gibberellin. And what the gibberellin does, some rhizobia get into nodules. They start making gibberellin. That stops the plant from making any more nodules. So the plant has to give everything it's got to those early settlers. And here, start out with a 50-50 mix of the two, and the gibberellin producers, come to dominate the root system over plant growth. Now, I couldn't actually find anything on the effect of this on the plant, but my assumption is that stopping the plant from making as many nodules as it thinks it needs probably is not a good thing from the standpoint of the plant. And yet, there's this vast literature of so-called plant growth promoting rhizobacteria. Bacteria that seem to have a beneficial effect on plant growth. Many of them are living on the root surface or nearby in the rhizosphere. And it's very strange because some of these bacteria do things the plant can't do for itself, like maybe solubilizing phosphorus or making a toxin that defends the plant against root pathogens, or nitrogen fixation, which is what I study. But there are an awful lot of examples where it seems that all these bacteria are doing is producing some hormone. You say, oh, hormones stimulates plant growth, but it only takes a few micromoles of hormone. It's very, very small quantities. So the metabolic cost saving of having bacteria make your hormones rather than making your cells is negligible. In fact, it's negative, because if you're a plant pumping carbon out into the rhizosphere to feed these hormone makers, most of that carbon is eaten by who knows what, pathogens or anything. So it just seems very strange. I want to review that literature sometime. But I suggested in this recent perspective that if you think about hormones made by rhizobia, there may be a few possibilities there. Because the rhizobia are in the root nodule, the plant's supporting them anyway, the carbon that goes to the root nodule isn't going to a bunch of other things, but still micromoles, it's a negligible cost. And so this is only going to be beneficial if the phenotypic change in the plant that's caused by these hormones is beneficial to the plant. It's not cost savings. Now, why might it be beneficial? Well, it could be beneficial if the rhizobia have more information about what plant phenotype is optimal for the plant in the current environment than the plant itself does. And in addition to that, you would have to have rhizobial fitness strongly aligned with plant fitness, because even if the rhizobia know what's best for the plant, they're going to do what's best for the rhizobia. So I suggested these two criteria would not be met very often. But I don't think it can rule it out entirely, because there are a lot of cases where the phenotype that's fitness optimizing for an individual plant in its competition with other plants is not the phenotype that maximizes community level benefits. I mean, when the Irish elk were going extinct because their antlers were too big, bull elk with bigger antlers still got all the mates. This principle of trade-offs between individual and community is the main theme of my book. It's not mostly about pesticides, resistance, or anything like that. Borlaug, I'm not sure how clearly he understood this, but Peter Jennings, who developed the Green Revolution rice, clearly did. He actually laid out in 1964 his plan, what kind of phenotype it would take to increase rice yield. He went and selected plants, crosses that gave that phenotype, doubled rice yield overnight. And he suspected that there would be trade-offs because his high yield plants were high yield partly because they were short, they were putting resources into grain rather than stem, and that meant they would be less competitive. He did the experiment, and sure enough, in competition with the older materials, his yield doubling materials disappeared over the course of 4 years. That was actually anticipated. Earlier work with barley by Sunison at UC Davis saw the same thing. High yield materials disappearing in competition with low yield materials. It does not mean that low yield materials, get this right, doesn't mean that low yield materials will always have higher fitness or vice versa. But it does suggest that the materials that are the genotypes that do best in the community probably are not the most competitive. Now, lots of examples in my book, but this is actually some old work of mine where they hypothesized that leaves that track the sun are going to get a little more light, a little more photosynthesis, but they also cast a larger shadow. And some computer modeling that I did suggested that this additional shading of lower leaves could actually result in lower photosynthesis from solar tracking on a community level, even though the individual leaves had higher photosynthesis. And we did some experiments where we disrupted solar tracking and measured photosynthesis and so on. And sure enough, there was a benefit of solar tracking at low leaf area where the shadows of the leaves were falling on soil. Well, once the shadows of leaves started falling on other leaves, you actually had a cost of solar tracking. And so the suggestion was that this trait has persisted despite its community level cost, because the shadows cast by those leaves are often falling on neighboring plants and suppressing competitors. Well, here's another example that getting to how we might use the rhizobia. We just had a drought, and so there's a lot of interest in drought-tolerant crops. And both Monsanto and Pioneer have developed what they called drought-tolerant maize. And in both cases, there's a really strange thing, which is if you look at soil moisture over weeks, the so-called drought-tolerant maize is sitting in wetter soil. It's not water limited, it's got water in the soil, whereas the Czech variety doesn't in both of these cases. Now you might suspect that, with $100 million on the line, somebody stuck out in the middle of the night and watered the drought-tolerant materials. But there is a more scientific explanation, and that is the reason there was water in the soil is that the so-called drought-tolerant materials had not used it. They had grown less relative to the Czech. They had less leaf area relative to the Czech, and therefore they left water in the soil for when it was really needed. Now think about this. The ancestors of maize, teosinte and so on, have had millions of years of exposure to drought. Why didn't natural selection come up with this? Well, because a plant that leaves water in the soil for later use, its neighbors could take that water, unless the whole field is the same genotype, which is what we have in agriculture. Well, what's the chance of exploiting this individual versus community trade-off in water use in a way that could increase crop yields without having to use more fertilizer or irrigation and not having to drain those wetlands and clear the tropical forests? Tom Sinclair, my major professor, who's still active at, he's older than I am, modeled this trait of limiting transpirational water use by soybean. Modeled it as a function of year and as a function of geography, and concluded that 70% of the time, this transpiration limiting trait would be beneficial to soybean yield. Now, Walid Sadak, a colleague in this building somewhere, looked at rhizobia. We know that rhizobia, some of them make auxin, plant hormone. So the question is that a way that we could achieve this goal of reducing transpiration in a beneficial way? And what he found, I think this is data for wheat, is that higher levels of root auxin resulted in substantially lower transpiration, which potentially could give this benefit. So even though in general, I think manipulation of plants by bacteria is likely to be harmful, if that manipulation essentially forces the plant to be more public spirited and expand community level benefits at the expense of that plant's competitiveness, then it might be useful. So to summarize, I suggested that we could reduce the amount of nitrogen, reduce fertilizer needs by breeding for lower protein maize, so that there's less nitrogen leaving in the maize grain that has to be replaced. We still need that protein, but that could come from nitrogen fixing soybeans, not from fertilizer. If we can use, if we can increase fixation without increasing its cost to the plant, possibly by developing cultivars that have stricter sanctions against mediocre rhizobia. And this is all fairly speculative. Well, this part is speculative, I would say. And then this last part's pretty speculative as well. There is evidence that legumes and rhizobia manipulate each other, but whether we could actually exploit that manipulation in a beneficial way, I would certainly call speculative. I'm just going to click through my bibliography so that people know it's in the recorded version of the talk, if anybody's interested in looking those up. And do I have time for questions?
Maybe so we've got 5 minutes for questions, and people who are online, maybe you could raise your hand and then I'll call on you, but we can start with folks in the room. Anybody in the room has questions? Mike.
So which would you advocate as being more potentially effective? Manipulating the working up primarily on plants, or maybe working on, making modifying their rhizobia and seeding the field with rhizobia that don't cheat, or whatever word you
generally what happens is you can inoculate a plant with whatever you want, but what ends up in the nodules is what's in the soil. There may be ways to get around that, but generally inoculation has not had it does not work. I mean, the first time a legume is grown, you can inoculate and that's what you'll get in the nodules. But the next year, you get the mutants of that initial inoculum, which are not necessarily as beneficial. So no, I don't think there's any point. The idea of the manipulating rhizobia could be an exception, because there might be a case where maybe you would only need 10% of the rhizobia in the plant to be auxin producers to get the phenotypic effect. And getting 10% is probably feasible, but getting 75%, I don't see any prospect of that happening.
Great. So sorry, Sarah raised her hand on Zoom, and then we'll come back. Sarah, if you could unmute and try stating your question, and we'll see if we could hear it in the room.
Okay, I had 2 related questions. One, can you hear me?
Yes.
Yes.
I don't think they can.
One question is, it seems like this depends on the extra nitrogen that's fixed being allocated to grain, as opposed to being allocated to leaves or roots, for example, because if you increase the N fixation rate and you just produce more N-rich residue, then that's going to promote N loss. So I was wondering if you could talk about the allocation issue. And then and then related to that, I was looking at your nitrogen budgets, and it looked like there's still the same amount of N being lost in the two scenarios. And so maybe you could clarify.
Yeah,
Okay.
so first, yes, the nitrogen fixed has to be allocated to grain. That's what happens in current soybeans. You know, a harvested soybean field, there's not much nitrogen there. It's almost all gone in the grain. And that would have to be maintained in the system that I propose. You're right that I didn't change the estimate of leaching in those graphs. If people are over applying nitrogen fertilizer, then cutting fertilizer use, if they're applying more fertilizer than the plant can take up, then cutting that is going to reduce leaching. But once you get down to where you're just supplying the nitrogen the plant actually needs, I'm not sure how much decrease in leaching we would get. I hope there would be some, but the big benefit of doing this, beyond all the other stuff that we're trying to do to cut leaching, is that if you're making half as much nitrogen fertilizer, there's half as much greenhouse gas being released in the process of making the fertilizer. That's a benefit I'm more sure of than I am of a decrease in leaching.
Thank you.
Great, thanks. So maybe one more, we'll take Naveen's question.
Can you go back to the summary slide, please? Can you go back a couple of slides to the summary slide?
Go back a couple of slides, can I? Should be possible, but it might take me a while. Any chance you could just ask about it?
I could. So one of the points in the summary slide was that the plants could sort of sanction against mediocre rhizobia.
Yes.
Right. And so my question here is, for an individual, obviously, it makes it if an individual plant sanctions against the mediocre rhizobia, isn't there a smaller pool of rhizobia it can form nodules with?
Oh, right. So his question is, if mediocre rhizobia get shut down by sanctions, will there be a smaller pool of rhizobia?
Right, and that leads me to my
Yes.
the following question. Which
Yes,
is that,
there will be, but you could cut the number of rhizobia in the soil by 90%, and still the plant would make just as many nodules.
okay.
There's a surplus.
Yeah. So I guess my question, the question following that was like, would an individual have less of? So if it was in a community, if there are multiple genotypes in the community, would it be a problem for an individual to sanction very strictly against mediocre genotypes? Would it have a competitive disadvantage?
Yes, it's quite possible that a plant, well, the reason that legumes don't shut down the mediocre rhizobia is that from the immediate short-term interests of that plant, those rhizobia are still worth supporting. And so all the neighboring plants are saying, please kill them. We don't want to have to deal with them next year. Yeah, well, you kill them. So, you know, it's another tragedy of the commons situation where it's not going to be solved by natural selection, but human imposed selection could do that. Now, that would probably mean that the first time we grew one of these soybeans, you'd have to put a little bit of fertilizer. And after that, you'd have good results.
Great. Well, I think we are out of time for more questions, but let's thank Ford one more time for that. Great.